This is a production of Cornell University.
Let me proceed. I'm not going to talk about photosynthesis much, but I'm going to talk about photosynthetic organisms and their lifestyle. And it's the what I'm going to talk about is this business, this yin and the yang that we have known for the longest time, which is that there are cells like cyanobacterial cells that can perform these two amazing and important energy-related processes, one producing energy and the other one utilizing energy, and that's photosynthesis and nitrogen fixation. And let me just start by saying something that probably everybody in this room knows, but this is the basis for this study, is that photosynthesis, especially oxygenic photosynthesis, produces oxygen. Nitrogen fixation depends on an enzyme that's exquisitely sensitive to oxygen. So having the two things happen in the same space, in the same time, or not, this has been a long time question that people have been asking. And what I will tell you now is what we have learned over the last 15 years or so studying specialized cyanobacterial cells. This is my favorite slide always to remind myself, and also when I teach students, is that oxygenic photosynthesis, nature devised this amazing system long time ago. It's carbon negative. It produces the byproduct oxygen that we all depend on, and it makes good stuff like these carbohydrates. That's the basis for life as we know on this planet. And the other thing that I want to point out is that the basic machinery for photosynthesis evolved in cyanobacteria as far as oxygenic photosynthesis is concerned, and remained the same over many millions years of, or billions of years of evolution, so that what we see in plant systems, the basic machinery of photosynthesis remains the same as what evolved into a cyanobacterial system. So what are cyanobacteria? I am assuming that majority of the folks here are plant biologists. So cyanobacteria are microbes. They are prokaryotes, oxygenic photosynthetic prokaryotes. They are the progenitors of chloroplasts. Most abundant fossils in Precambrian rocks. They have been around for many billion years and found everywhere on the planet. What is going to be important today is this feature that they are the only known bacteria with circadian lifestyles. And this will play an important role in what I'm going to discuss today. Now, cyanobacteria, I'm just going to quote Stephen Jay Gould's Book of Life from that. It says that the 2.5 to 2 billion years ago, cyanobacteria thrived by using water, the most abundant source of hydrogen in the environment, and oxygen was a byproduct. But here is the important sentence. Life began to change the planet. The planet the way we know it, the oxic environment, primarily the cyanobacteria are responsible for it. One of the consequence was this business of all iron 2 plus becoming iron 3 plus, precipitated to the ocean bottom. Basically, for biology, this is a big conundrum because the basic machinery of life was dependent on iron 2 plus, but then all of a sudden we don't have any of that, and everybody, including human beings, we have to invent all sorts of ways of capturing iron and using it at the appropriate level. And then nearly over a billion years, oxygen level built up, dissolved in water, is kept in the atmosphere, 20.6%. Sometimes it was even higher. And stage was set for the evolution of eukaryotes. Without this, this probably would not have happened. So Gould says tiny organisms that exploited the wealth of CO2, water, and sunlight transformed the earth. So these are cyanobacteria. They look like this, wide variety of habitats. They're morphologically diverse, and some can also fix nitrogen. What I show here, those are supposed to be micron. That's the Mac to PC conversion. So they can be very large, like 10 micron bacterial cells, and unicellular, whereas they can also be in filamentous phase, like in Anabina, a system that has been studied. The three in the bottom are the one that can fix nitrogen. Synechocystis 6803, that's a workhorse that we have been working on for more than 30 years now. That does not know how to fix nitrogen. I'm going to compare in my this one with that one, cyano-TC 51142 and 6803, those two numbers that will come up over and over again, and you will know what I'm talking about. This one cannot fix nitrogen, this one can, but both of those are photosynthetic, oxygenic photosynthetic organisms. So let's go into cyano-TC. What I'm going to describe to you is a project that has been ongoing for more than 15 years now with this class of organisms. Now, let me go back and point out something. When nitrogen fixation takes place in a photosynthetic organism, as I pointed out, one has to separate out the two processes, the process of oxygen production and the process of nitrogen fixation, because the two are the yin and the yang, as I mentioned. Now, organisms have developed two different strategies to do that. It's easy to understand. One is over space, and that's this kind of filament. The cells that are smaller, those are the vegetative cells where photosynthesis is taking place. The cells that are larger are the heterocyst cells where nitrogen fixation is taking place. So the two kinds of cells and elaborate developmental processes that are taking place has been very well studied over the years. This is separating out the two processes in space. In contrast, what you will see is that these kinds of single cell organisms that fix nitrogen, single cell cyanobacteria, they separate out the processes in time. And this is of interest here. And that's where the circadian rhythm business comes into play. Okay, so cyano-TC, again, here are the different kinds of cyano-TC. I'm not going to spend a lot of time. They have been isolated from various parts of the world. The one that we work on primarily, 51142, that was isolated by Lou Sherman's lab at Purdue when they went out to Port Aransas in Texas. So these are from benthic water. And they are also found in open ocean, and very interesting stories that are developing in how those cyano-TCs operate in the open ocean. Okay. Now, what I want to point out here is that it's the phylogenetic relationship of cyano-TC. Nowadays, it's easy to sequence a genome. Any given evening, you can sequence a bacterial genome, and you have tons and tons of data, and then you run them through pipelines, and then you find out all kinds of things. This is some years ago in 2011. Here are a majority of the ones that were known. And what I show here is that cyano-TC, again, that 51142. And here is the other one that does not know how to fix nitrogen. What should be very clear here is that the two are very closely related in this whole scheme of the things. And I'll come back to this notion. In contrast, there are other cyanobacterial species like this Synechococcus. They are quite distant from a phylogenetic point of view. Okay? And this will play an important role. So let's talk about 51142 and what is an approach that's is being called nowadays systems biology, which is looking at all things that happen in the cells. We used to call this thing physiology in the past. So that's what we are going to go into, but in much more detail and with an approach that takes into account the entire genome and its associated functions. Okay, so it's unicellular and fixes nitrogen. Diurnal rhythm results in numerous intracellular changes. And in this electron micrograph, what I want to show you is what a cyanobacterial cell looks like. It's a typical gram-negative bacterium, has an outer envelope layer with the plasma membrane and outer membrane. And inside is this whole elaborate network of membranes. That's the thylakoid membranes. That's the same kind of membrane system that's present in chloroplasts. And then it has all kinds of other things, intracellular body. One that is being studied at this institution very much is this one called carboxysome, where carbon fixation takes place and so on. Okay, so let me first show you the cellular ultrastructure. Michelle Liverton, when as a graduate student, did this study. And it's a 2 nanometer tomographic slice images that show the intracellular details, showing the thylakoid membranes, how they are laid down. And G here is glycogen bodies or glycogen storage bodies. C is carboxysome. And so all of these are shown, and these are individual slices shown one after another. When you put those two together, this is what you see. There is a movie associated with it, but I will not go into the complication. It's again Mac to PC conversion. But what you see here is this elaborate description. This is still the largest or the most detailed description of a cyanobacterial cell to date. Basically, what it shows is the entire cell. This is a cutout, and it's full with those green-colored, pseudo-colored membrane system. That's the thylakoid membrane. So if you look at that cell, a typical E. coli cell and a typical cyanobacterial cell, that will be the distinguishing feature, the intracellular membrane system, that's thylakoid. Also, we counted numbers, so there are 21 carboxysomes and so on and so forth. Okay? All right, so this is what a cyanobacterial cell looks like. Okay, so now let's go into the business of dynamics or rhythms. What happens to these cells under alternating light-dark conditions? I'm going to stay away from considerations of circadian rhythm. That's a set of studies that I'm not going to talk about. There are some subtle differences between diurnal rhythms and circadian rhythm studies. But if anybody is interested, I'll be happy to discuss that later on. So diurnal rhythms, we set up experiments that are very, very simple. It requires 12 hours of light. That's the L side, the left side, and 12 hours of darkness. That's the blue side on the right side. And basically, the first thing to observe is that when we look at specific time points during the light period and the dark period, and we measure photosynthesis, here is darkness, here is light, and what you see is that the capacity of photosynthesis goes down dramatically during the dark period, and it comes back up during the light period. And in contrast, nitrogen fixation takes place exclusively in the dark period. Okay? That's the temporal separation of the two processes. So basically what we did in those early days is take samples every 2 hours, which means that there are a whole bunch of undergraduate students who lose their circadian rhythms, and they collect all of these samples. And what was interesting is that this system is so robust, is that looking at the data, you can immediately say, okay, this person at 2 o'clock in the morning did not take the right sample because of the oscillations that take place in various things that we saw. So anyway, so collecting those samples, and those samples were then used in a variety of, the same samples were used in a variety of omics level studies, so to say. But first, let me show you what happens to the organization of the thylakoid between light and dark. This is in darkness, D10, that's 10 hours into darkness. This is L10, this is light, 10 hours. And what you see is that here are the thylakoid membranes. Their relative orientations change, but two different kinds of intracellular bodies that are present. Here is a glycogen granule that accumulate during the light phase, because that's where active photosynthesis is taking place, and the carbon is getting stored in the form of glycogen. In the dark phase, as you will see in a few minutes, that glycogen is broken down. That's actually the engine that drives nitrogen fixation. And the nitrogen that's fixed goes into these bodies that are called cyanophysin granules, and that's the storage area for, or the volume for the fixed nitrogen, which is subsequently used in biosynthesis during the following day. So it's going over and over again through these processes in a periodic diurnal manner. Okay? So it's a beautiful system, and as I have mentioned, photosynthesis and nitrogen fixation are separated temporally in the same cell. Okay, so now comes into looking at all different kinds of molecules. I'm going to spend a lot of time on transcriptomics, but we have done transcript, protein, metabolite, post-translational modifications, all of those, but basically with the same samples so that we can compare the different omics level data one with the other. So let's talk about transcriptomics. Well, the first thing is the nitrogenase gene cluster. Nitrogenase, this enzyme, is large. It requires a few proteins that are components of the nitrogenase enzymes, but it also requires all sorts of very unusual cofactors, the details of which we are beginning to understand. And to synthesize and insert all of those cofactors into nitrogenase, there is a large set of genes that are necessary. And in many bacteria, by the way, bacteria and archaea are the only organisms, only prokaryotes can fix nitrogen. There is no eukaryote that's known that can fix nitrogen. So in cyano-TC51142, there is this large set of contiguous genes in gene clusters, a whole set that's looking leftward and a whole set that's looking rightward. altogether 35 genes. I will come back to this business of 35 genes. And different cyanotheses have practically the same type of organization of the NIF cluster. Okay, so let's first look at the expression profiles at the transcript level of all of these genes. And what we found is really very tight coordinate control of the expression at the transcriptional level. Let me describe this to you. This is a log-2 ratio, and there is a 9 log difference between the top of that and the bottom of that. Okay, this is dark period, this is light period, again dark, and this is light. Okay, so all of these genes are getting coordinately controlled, coming all from this cluster of this 28,000 base pair chunk of DNA that's present in cyanothece, and you can see how tightly they are controlled. We have continued these experiments in subsequent years for many, many days, and that does not die away. So it's very carefully controlled. Okay, now if we take all of the transcriptome data and we do modeling on those by doing very simple Pearson correlation coefficients and looking at what are the different genes that have the same kind of expression profile over a diurnal behavior, then we came up with this kind of a model that we call this a Reed model. Okay. Let me describe this. These are different time points. Dark 1 hour, 5 hour, 9 hours. This is light 1 hour, 5 hour, 9 hours. Red is upregulation 4 times, and this faint blue color is 16 times downregulation. Okay. Now let's look at this area. All of these actually show all the genes that are present in a cyanobacterium, in this case the cyanothece 51142. So every dot represents the transcription level for every gene. Okay, and that's invariant in this structure. What is variant are the levels of expression. So in the darkness, what I say energy metabolism, basically respiration, that's coming up first, followed by nitrogen fixation. But they also go down toward the end of the dark period. What comes up is this region, that's photosynthesis. The lights, it's lighting up, and it's lighting up really high here. But that also goes down during the light period toward the latter part of the light period. So this is not just the presence of light or darkness, but a careful programming inside the cell that determines the transcriptional profiles of every gene that's present in this organism. Okay. And based on those data, this is only based on the transcriptional profile, I want to emphasize that, we came up with this core cellular model of the processes that are taking place inside the cyanobacterial cell. And it's, again, very simple in this diagram. On the left, I show all the things that are happening in the light. This is summing up the metabolic processes that are taking place. And in the right part are all the things that are happening in the darkness. Let me describe this to you. Even though this is a photosynthetic organism, this bug actually is very active also in the darkness. That's the bottom line of this thing. As one expects, photosynthesis, meaning both the photo part, the electron transfer processes, as well as the synthesis part, the carbon fixation processes, are extraordinarily highly active in this part. The carbon gets fixed and stored in glycogen, as I mentioned. And in the darkness then, the glycogen is degraded, the glycogen degradation pathway turns on, and then respiration comes up at a very high level. That takes away all the oxygen from inside the cells. We have direct measurements of these. And then nitrogen fixation takes place. But it's not just nitrogen fixation. Many, many processes, such as hydrogen production, hydrogenase, things like that, are also controlled in the same way. So in this period of time, the inside of the cell is relatively anoxic. And that's the unusual and special feature of this kind of cyanobacterial cells. And then amino acid biosynthesis happens here. The extra nitrogen is stored away in cyanophycin, and it goes back. And this continues to happen over and over and over again. So the diurnal cycle, summarizing that, photosynthesis results in oxygen production during the day. High rates of respiration generate low intracellular oxygen tension during early night hours. And oxygen-sensitive enzymes such as nitrogenase can then function in the same cell. Okay. Since Klaus is my host, I thought that I should at least touch on the next topic, which is proteomics. As I said, we have done this interrogation at different levels. Now, we all know, after many years of systems biology, that mapping one omics level map onto another omics level map still remains a challenging thing. And that's simply because from very basic biology, there is no reason to believe that what happens at the transcription level will be essentially followed at the level of all the post-transcriptional, translational, and post-translational events. So we were interested in seeing what is the level of correlation between the diurnal oscillations, or if there is any diurnal oscillation. So same kind of experiments. It required a little bit more time. We did these experiments in collaboration with Dick Smith and colleagues at PNNL, and in those days, these were really challenging experiments. It kind of tells you what we have done, but basically the same thing. Every 2 hours, we collected samples, the same samples that were used for transcriptomics, and this is what the cycling patterns of the proteins look like. Same kind of color scheme, okay? So the first thing to notice is that the read is not that dense. So not that many proteins at the level of detection that we had are oscillating in their absolute protein levels. But the overall story is the same. Again, photosynthesis comes up during light, and nitrogen fixation comes up in the dark time. And here is a description of what happens at an overall protein level. And we did this experiment. This was, at that time, a super expensive experiment to do all these things over all these time points. But we decided to do it at least for 2 cycles, just to see what is repeatable. And just by looking at the profile, you can see that the same kind of profile, more or less, is emerging day after day. And so 251 proteins exhibited measurable oscillations in their abundance under alternating light-dark conditions. Who are they? We go in through there, and basically, without looking at the details, basically 25% of them belong to central intermediary metabolic pathway, and 19% are involved in photosynthesis and respiration. These are proteins whose absolute amounts are changing over the light-dark cycle. Okay? But let's go back to nitrogenase now. And here is, again, something really, really interesting. Here are the transcript levels of all of the genes. I show only the structural genes, NIF, HDK, and 2 other genes, just to make it simple, and see how they have very tightly controlled a single peak in the middle of the dark period. But during the dark period, it also goes down. And I'm emphasizing this because there is a story behind that we do not have much idea about it, but biology does it this way. It does not, there is a program that determines that things will come up during a period of time, and then things will go down during the same apparent dark period or light period. Here is what happens with the proteins. And as you can see, the proteins are also getting degraded. They get synthesized, but they get degraded during the night time, and their levels are extraordinarily low in the light period. Also, there are binary oscillation profiles. If anybody is interested, we can talk about that. Those are interesting stories by themselves. So during the cyanotherapy diurnal cycle, the regulation of nitrogenase, the NIF genes are expressed during the dark period, NIF structural proteins are synthesized during the dark period. We have done dynamic proteomics, which shows that the NIF proteins are degraded during late dark hours. And in those studies, what we find is that post-translational modifications are important. I don't have time to go through that. We'll be happy to discuss that later on with you. And nitrogenase assembles and functions during early and middle dark hours. Okay, now comes the Yang. Remember, the same cell is also photosynthesizing. And so the question that we asked, is it just darkness that turns off photosynthesis? Or are there extra exquisite control elements that are present in the organism that makes sure that oxygen is not evolved when nitrogenase is functioning? And what I will tell you is, again, a snapshot of only one of many things that are happening exactly in that way, where there is cellular control in protecting the nitrogenase from accidental production of oxygen from photosystem II. That's the oxygen producer. So in photosynthesis, photosystem II is the enzyme that produces oxygen. We know a lot about this system. Class just just reminded me of those early days when we did not even know how to isolate these things. And then over the years, it has gone to a stage where we have a 1.9 angstrom resolution structure that looks like this. And all I can say is that PS2 complex is extremely complex. It has lots of proteins, and it has also lots of cofactors. The ones that are important here that I'm pointing out here are 4 manganese ions. We have known from early studies with cyanobacterial cells with people like George Chenet from the 1960s, where they have looked at how manganese gets inserted in photosystem II, how that gets functional, and so on and so forth. Now, the reason I pointed out manganese is that at the center, this is a cartoon diagram that simplifies that very complex structure. At the heart of photosystem II are these 2 proteins, D1 and D2. D1 is the protein that plays important role in coordinating those manganese atoms in photosystem II, and those manganese atoms are absolutely necessary for the oxygen evolution activity of photosystem II. Okay, so long story short, if photosystem II cannot bind manganese, you take away the possibility of oxygen production by photosystem II if there is a little bit of light around, okay? Because a little bit of oxygen is enough to kill nitrogenase. So D1 protein that's encoded by this gene called PSBA, it's a well-known gene, provides many critical ligands for the manganese cluster that is at the heart of water splitting and oxygen evolution. PSBA has 5 copies in cyano-TC 51142. And out of these 5, only one that we call PSBA4, that's just a nomenclature. All of the other 4 are expressed highly during the light period, which is where they function. Okay, so here is light, and those red lines actually shows the sum total of all the 4 genes of how they behave at the transcriptional level. We notice there is a 5th copy, this PSBA4, that's behaving just the opposite. Okay, the red one is the one that's PSBA4. The other ones are, the black ones are the ones that are the normal PSBA. So we said, this is kind of strange. Why is a D1 protein being expressed in the darkness? It has no business of being expressed at that time because photosystem II is non-functional at that time. It became clear when we sequenced the gene and the protein part. There are specific amino acid residues that we know from that crystal structure that are involved in ligation of the manganese cluster. And nature has done that beautiful site-specific mutagenesis experiment many billion years ago in cyano-TC, where each and every one of those residues are taken out and replaced by things that in the 1980s and 90s, many of us were involved in doing site-specific mutagenesis to find out which residue change gives rise to non-ligation of manganese. So long story short, we recapitulated this in synechocystis by taking this gene from cyano-TC, PSBA4, expressing it in a synechocystis 6803 cell that does not have any PSBA gene. And what we find is that that one, the one shown with red, cannot, PS2 is formed, but it cannot produce oxygen. This is expected, but we show it this way. Whereas if we take one of those other genes, the PSBA1, then introduction of that into synechocystis allows photosystem II to function. So what I'm telling you here is that here is again a control system. Now, why do you need in the darkness? And then the other thing that I must mention, that when we looked at this specific form of PSBA that has these amino acid replacements, they are present in many diazotrophic cyanobacterial strains. Okay? And the list has grown tremendously since we published this thing. And we call it the Sentinel D1 because it is a way of the cells making sure that oxygen is not evolved. Now, why is this important? And this was something that was discovered by Kim Wagner, who was a former graduate student, now works at a local company. She said, well, what's the intensity of moonlight in a full moon night, and cyanobacteria are growing on the surface of the pond, and did the simulation in the lab, and basically there is enough oxygen there that kills nitrogenase activity in cyano-TC under those conditions. Is that the real natural reasons? We don't know. But that's one of the reasons probably the cyanobacteria go to this extreme control systems where they make sure that photosystem II is not is functional at night time by putting in this strange copy of D1 protein that cannot function as a manganese ligation protein. So we envision, we have some proof but not definitive proof of this entire model. We are working on that. Basically in the darkness, dissociation of some other proteins and synthesis and insertion of SD1, that's the sentinel D1, and then removal of SD1 and insertion of D1 when light comes up. Okay? These are challenging experiments, but we have initial evidence that this is the way cyanothesis is operating. So this is the yin and the yang. How much time do I have? Do I have about 5 minutes or so? Okay, all right. So let me just go into the next phase of another challenging set of experiments that we have embarked on. Now it has been now close to 3 years now that we have been involved in this. And this is the project has to do with designing nitrogen fixing ability in oxygenic photosynthetic cells. And the background that I provided to you with the cyanothesis and synecocystis systems will make it easier for you to understand where we want to go with this. This is an evolving story. It's a collaborative one where Kostas Maranas is a computational scientist at Penn State, and then two of my synthetic biology colleagues in the engineering school of Washington University. We have teamed up to do this. Why even get into this? That's because nitrogen is an essential nutrient for plant growth, and a long-term goal of this nitrogen program is to endow plant cells with nitrogen fixing ability. This is the big goal that a lot of people are interested in. And as I mentioned, photosynthesis in plants produce oxygen, a potent inhibitor of nitrogenase. But on the other hand, nitrogenase needs a lot of energy. So you cannot put nitrogenase in a tissue that does not have enough energy source. It will not be able to fix nitrogen. So how do you deal with this? So the overall goal for our systems and synthetic biology approaches is to develop the design principles to establish nitrogen fixing ability in the unicellular cyanobacterium Synecocystis 6803. And the knowledge base that's created in this project will be directly applicable to engineer nitrogen fixing crop plants, we hope. Okay. All right. So this is kind of a schematic diagram of what we are talking about. We want to make a nitrogen fixing crop plant. There are two ways to do that. One is to engineer nitrogen fixing symbiosis for crop plants, just like soybean does it. The other one is putting nitrogen fixing module directly into the photosynthetic tissues of the plant. Okay, so one way to think about it is that we just heard all these things about cyanothesis 51142, just transfer all that entire machinery into plants. This is challenging. So we did not even approach this. Instead, we said, let's just take a shortcut and let's try to figure out the principles that are involved in putting in nitrogenase into an oxygenic environment in an organism that does not know how to fix nitrogen. So we go to our good old friend Synecocystis, and we want to transfer the nitrogenase program, not just the genes and proteins, but the program itself, all of it, into Synecocystis, make Synecocystis that's diazotrophic, and then the design principles, presumably in some future years, will be used for making this nitrogen fixing plant. That's the basic thing. Okay, so we chose these two organisms simply again, bringing back that they're phylogenetically very closely related. In many ways, Synecocystis is a nitrogen-fixing cyanobacterium which has lost its ability to fix nitrogen. That's the way people have described it in papers that describe the phylogenetic relationship. And our goal is to establish in Synecocystis the nitrogen fixation circuitry normally present in cyanothesis. What I'm going to tell you in 2 minutes is that the first phase of it is over, because we have been able to now, this was the biggest challenge at the beginning of the project, is this 35 genes in NIF cluster, put all of these in, move them from cyanothesis into Synecocystis. And there are new techniques that are used by the synthetic biologists that allows us to do this. So we could use a method called DNA assembler that, again, the details are not important here, but if anybody's interested, I will be happy to discuss that. And very quickly, first of all, put together an E. coli, first in yeast and then in E. coli, this large plasmid that's about 40 kilobase pair, that has all the genes here exactly in the way cyanotc has it. We decided to take this approach rather than the traditional synthetic biology approaches that Chris Voigt and colleagues are doing, which is first taking apart every gene, putting in control circuits on them, and putting it back together and make it work. We thought that we are not clever enough to do that, so just borrow the whole thing from a cyanobacterial cells, move it into a cyanobacterial cells. It took a little bit of time, but we could transfer all of this into Synechocystis. And for the last 2 years, this thing is stably replicating in Synechocystis. Growth is not compromised because of the production of all of these genes. And we know at the transcriptional level, all of these gene products are made, all of these 35. I also show you another construct where we have now started whittling down the 35, and we have now down to 24, and we have it down now to even 10. I'm not going to show you those data. But basically, the stage that we are right now is that Synechocystis 6803 can fix nitrogen. So basically what it shows here is that here are the 3 genes that make the 3 structural proteins of nitrogenase, NIF-HD-K. In plants, only one of these, the iron protein, has been expressed in chloroplasts with great difficulty because of, again, this bad thing called oxygen. So at every level of the production of the functional nitrogenase, there is oxygen control in a negative manner. And what we see here is that in this organism where we put in all of this, we can have transcription of NIF-HD-K. The black one actually shows the whole set of 35 genes. And I show you this truncated version 24, which has appreciably higher level of transcription level for all of these. So there are some negative regulators here, and I will not go into the details of that. But important thing is this. This is the first time, to our knowledge, that in an oxygenic photosynthetic organism, one can establish the nitrogen fixation machinery. And so you could see that there is production that's taking place, nitrogen fixation. Let me also point out, because usually this question comes up, this organism is still not diazotrophic because the control circuits are not appropriate. In fact, this bug, the Synechocystis that has these nitrogenase genes, it produces the nitrogenase at the wrong time, namely at the beginning of the day, because that's the normal program inside the cells. We have had recent success in providing some control elements in this cell to make it go the way cyanotc goes, but we are still quite some distance from achieving the stage where a robust nitrogen fixation cycle, the diurnal rhythm, is expressed in cyanotc. This project is the first part of the project that I talked about on cyanotc systems biology is a collaboration with Lou Sherman at Purdue University and a whole bunch of people led by Dick Smith at PNNL, Pacific Northwest National Lab. And then for the nitrogen fixation project in Synechocystis, these are the 4 groups and all the people who are working on that. Here is my research group, and Eric Richards may still remember Ripstock Hall. It's right in front of our lab. And I thank you, and I thank you, my close collaborators, these cyanobacterial cells. I'll take questions.
Questions? It was a beautiful seminar. And what I'd like to ask, and I would like to ask you a question. In the beginning, you showed us the diameters of the different cyanobacteria. And the Synechocystis was smaller than the ones that do all the great stuff that you were talking about. Does that volume difference have any ramifications in getting the diurnal nitrogen fixation going?
The simple answer is we don't know. We have looked very hard for cells that are much smaller. There are cyanotcs that have been found principally by John Zayer's group at Santa Cruz that have much smaller size. But those are very special cyanotcs that do not have photosystem II. and carbon fixation liquidity. They are cells that live together with an algae. And there the story is more, the algae fixes carbon, and that specialized cyanobacteria produces fixes nitrogen. In that case, the cells are much smaller. So that will be a correlation. We have no idea, but we cannot address that question.
Yeah, so there are a lot of really interesting examples of regulation of things happening in the dark or light that started before the next phase actually occurred. Is there a readout of the nutrient state in the cell, and somehow nitrogenase is starting to shut off when glycogen is running out? Have you measured the glycogen at different points at night?
Yes and yes.
Yeah.
Yes, so glycogen runs out, but it's not strictly correlated with just the level of glycogen.
Mm-hmm.
Okay. Basically, it all comes down to the central control element that's found in bacteria, that's P2 protein in bacteria, that senses the relative levels of carbon and nitrogen.
Mm-hmm.
All of that story works fine here. But what is not known is how the central oscillator of cyanobacterial cells, which is very well defined through the work of Susan Gordon and colleagues called Chi proteins, Chi ABC, how is that controlling this entire program? And we don't have an answer to that, but some of those specialized regulatory elements are involved in figuring out the entire program. This is a complex program.
Mm-hmm.
A lot of oscillating systems have negative feedback loops. And I'm just wondering if you could elaborate on evidence for or against such a thing going on. So in other words, the synthesis of an enzyme reaches a certain concentration that then begins to shut down the synthesis.
At that level, again, we don't have any understanding at this level for this system. At the level of transcription, there is a lot of evidence. So there are negative feedbacks that are set up as well as positive feedbacks. Those regulatory elements have been identified by us and others. And so one of the places where we know quite a bit is this business of how the level of the transcript, I'm not talking about the proteins, of nitrogenase go down during the dark phase. It has to do with elements present in the genome that give rise to small antisense RNAs that get expressed exactly at that level. And we haven't proven it completely, but the idea is that then gets turned on. This whole business of, in this kind of rhythm, this whole question of anticipation is a big one. And we see evidence of that in every step of the way. The cell knows when light comes on, so they get prepared for that by synthesizing both at the transcript protein cofactor level of all the photosystems. The cells know that oxygen will be coming out at that time, so nitrogenase it goes down, and the other way also. I believe that there will be many such control elements that are present, but we do not have much idea. But as far as the enzyme levels and the feedback on that, not much.
Just one follow-up. Have you kind of duplicated some of these experiments where you've extended the nighttime or the daylight period?
Yes. I guess we were talking about some of those. We have done all of these crazy experiments, 18.6 and 24. I can tell you if you go 20 hours of light and 4 hours of darkness, this thing doesn't work. And it definitely doesn't work when it's 4 hours of light and 20 hours of darkness. There is a range in which it works. Basically, I can summarize those studies by saying the following. As long as you have sufficient amount of glycogen that's being stored, then nitrogen fixation does take place. But in doing so, we also figured out that we can separate out the two central processes, respiration and nitrogen fixation during the dark period, quite a bit or much closer, depending on the length of the light in the dark period. So those are the classic time series experiments that we're doing and modeling on those. but it's complex.
It has ecological implications, doesn't it?
Absolutely, absolutely, yes.
Maybe related to that, part of that is, of course, time. It takes time to make a transcript, it takes time to integrate it, but it's also energy balance. If you have to synthesize and remove your machineries, which is very costly, there's an energy cost. So
Right.
if you play, instead of this light-dark theory,
Right.
you play with the amount of light that you get, for instance, and you just titrate down the amount of energy it can accumulate. Do you see similar?
Well, not in great detail, but we have measured ATP and NADPH levels in these cells, and those change predictably, as you would expect, based on the energy demand. But as far as, what is the cause and what is the effect, very little idea so far.
Yeah.
Yeah, you showed that there's an ortholog of the Sentinel D1, or whatever it was, D1, yeah, in other cyanobacteria. Is that also found in the lineage loop in chloroplasts? And if so, what became of that when it is symbiosis?
Yeah, well, it's hard to tell what the exact lineage is, so I can't answer that question. But we correlate that with cyanobacteria that can fix nitrogen, as opposed to the ones that cannot fix nitrogen. That's the only correlation.
So how much conservation of those variant amino acids is there across the group that has this Sentinel D1?
Yeah, it's about...
So are they functional? Or
Right.
Some other function?
Actually, let's say we take a specific aspartic acid. Through site-specific immunogenicity experiments done in lab, we know that aspartate, if it is changed to an uncharged recipe, or even when it's changed to a glutamate, that compromises manganese ligation in interesting ways, and we have detailed information on that. What we find is that those changes are different in the different SD1. So it's more like nature took the canonical PSDA that makes the normal D1 and then made those specific changes so that it no longer can ligate manganese. But they are not the same changes.
What do you think is the center for the bacteria for the bacteria switching from the native protein, functional native protein to degradation? Do you think it's a protein or chemicals?
Again, my answer is we have no idea. It doesn't have to be that way, right? It could be that the proteins are made and they just stay around. And what's the need for making them? So, one phrase that I've used many times is that the first hour of darkness is the beginning of light for nitrogenase, right? Every day that's what happens. Transcripts get made, and then the transcripts get degraded, proteins get made, assembled, function happens. But they get degraded. And they get degraded at times when there is no oxygen. Why that is necessary, and whether that is absolutely necessary in the circuits that we are putting in systems, we don't have an answer, but we're trying to answer that. So I really don't have any idea of why that proteolysis is necessary.
You probably answered for me now, but can you tell us anything about any tagging systems or post-translation modification like arginine phosphorylation that was just discovered in the area?
Right. Arginine phosphorylation does play a role, but again, we don't know the extent of it.
Maybe one or two more questions.
One more, yeah. So globins play an important role in providing the noxic environment and linking nodules. Are globins expressed at all?
No, there's only
backup
one.
system?
Then
Maybe you
only,
can repeat the question.
yeah, so that globins that bind, like hemoglobin that binds, or myoglobin that binds oxygen, in the Ying system that I described, they are used to actually sequester the oxygen so that the local concentration of oxygen goes down. In Cyanobacteria, there is only one globin gene, and that has nothing to do with this business, similar for Cyanobacteria. So that's not the mechanism.
I was thinking maybe the backup to
No, that doesn't seem to be. And the absolute levels of those are very, very low. That cannot account for the local concentration of oxygen when even one photosystem II is working is super high. And so you really have to scrub it. And respiration does that job.
Okay, Alvin?
I'm just wondering if is there a specific machinery in Cyanobacteria for harvesting nitrogen that that the other bacteria do not have. So how does it harvest nitrogen?
Okay, so you are talking about already fixed nitrogen, like nitrate or ammonia. How does it do that? It's the standard thing, that chloroplasts have, which is transporters that are present, that bring them in, they are getting utilized. In fact, something that I didn't even show here or pointed out is that the organisms that fix nitrogen, they actually sense that very, very well. So if we put in even micromolar concentration of nitrate into the cyanophysis cells, nitrogen fixation ability goes down over a matter of 1 or 2 hours. So NIF-HDK synthesis stops, and again the proteins get degraded. So we don't know why that happens. But when there is enough fixed nitrogen, the organisms do not do this energy intensive. This is well known from work on other bacterial systems. Okay, I think we should end here. Thank you very much for the seminar.
This has been a production of Cornell University, on the web at cornell.edu.